So I would like to welcome Bart Eggen to stage. Bart is professor of molecular neurobiology at the University Medical Center in Groningen. And he is specialized in glial identity and how to identify them with all kinds of spatial omics technologies. So I would like to welcome Bart to the stage to give his lecture.
Cool. All right. Thanks, Mario, for the introduction, and thanks to organizers for having me here. So I'll share some of the data that we've been generating over the last few years, and I'll end up with roughly the last 15 minutes or so of unpublished data. So the good news is that I had some overlapping slides to pitch ICNS, which I will skip, so then I have more time to dive into data. So I work in the UMCG in Groningen, but I'm also affiliated with the RUG. That's where my chair is, and I work in the ICNS, but also in the Multiple Sclerosis Center in Noord-Nederland. So this is ICNS. I will skip this. But I think this is the most important point. My role in this project is that I like transcriptomics. I also like other omics, but I also like transcriptomics, so we do that a lot. And we analyze the data, and later in the program, Inge Holtman, who is also an ICNS in the UMCG, will talk about the bioinformatics and AI angle that we also use to analyze this data. So the brain, that's what I work on. It's a very complicated network containing neurons and glial cells, and the neurons are roughly half of the cells in your brain, and glial cells are the other half. And of that cell population, microglia is 10%. So 10% of your cells in your brain are microglia, and they're quite peculiar, and I will focus on these cells primarily for the next 20 minutes or so. Microglia are born in the yolk sac, which is a structure outside of the developing embryo, and then in humans, after 4.5 days, they go through the bloodstream, colonize the brain, and then they form a local resident population of macrophages, and they're self-sustained. The rest of your brain is coming from the ectoderm, but microglia are born outside of the developing embryo. They were first reported by Pío del Río Ortega. He was a student of Ramón y Cajal, the one that is famous for silver stains of neurons. So he published now 100 years ago, he published on microglia, and these illustrations you can see on the left, and on the right is a staining that we do in the lab using an antibody against a cell surface molecule, IBA1, and you can see that he already really carefully identified the structure and morphology of these cells, and he already predicted that they are phagocytes and that they are of myeloid origin, which was surprisingly correct, just by looking at these cells. So this is 100 years ago. These cells have then been increasingly investigated, and these cells are basically tissue-resident macrophages, so they have macrophage functions, say they phagocytose, they can mount an inflammatory response, but on top of that, they have a whole bunch of CNS-tailored properties. So they are important for myelination, the process that axons are ensheathed by a myelin sheath, which is required for action potential propagation. They regulate the permeability of the blood-brain barrier. They are important for vascularization. They remodel synaptic networks. So they are both typical macrophages, but on top of that, they have a whole lot of CNS-specific properties. And you can study that by using different approaches. And these microglia come in different flavors. And these flavors are basically determined by intrinsic factors. And intrinsic, I mean, what species are you, what sex, what age, what genotype are you made of. So these are all intrinsic factors that determine how what state a microglia is. And on top of that, it matters if you look at age. It matters if you look at region. So let's say microglia in gray matter or white matter or spinal cord are not the same. And what also matters is their history. So what have they encountered in their lifespan? So what did they see? Did they see infections? Did they see neurodegeneration? Did they see bacteria? All these things matter. I will not talk about this too much, but microglia have an innate immune memory. They have a memory where sort of external triggers land in their epigenome that they remember, and that determines their future responses to challenges. And the reason for that is that microglia are relatively long-lived. In humans, on average, the lifespan of a single microglia is between 2 and 10 years, depending on the brain region. So these are slowly cycling cells. So if things happen to them, there's an epigenetic imprint, and that determines their future responses. I like omics. We also do other things. We do proteomics, and we do morphometrics, and this and that. But for today, I will primarily talk about how we use transcriptomics to get an insight in microglia, how they change in the context of disease. And this toolbox that I will give you examples of is exactly the same toolbox that we are at the moment rolling out over psychiatric cases. So we are profiling many different brain regions from many different donors using a whole platform of omics approaches to figure out, as Mario indicated, if we can find gene signatures or maybe at the end of the day molecular drivers of psychiatric symptoms or psychiatric diseases or psychiatric spectra. Anyway, I like to use this cartoon to illustrate how we do this. So we get a piece of brain tissue in the lab. We get it typically from the Netherlands Brain Bank. And then we roll out, depending on what we want to do, different types of approaches to profile these cells. We can sometimes do a bulk profile, where we just take RNA out of a chunk of tissue, or we sort out our favorite cell type, or we do single cell or single nucleus RNA sequencing, or we do spatial, or combinations thereof. And then at the end of the day, we integrate that, and I will show you examples. For today, I decided just to focus on single nucleus RNA sequencing and spatial omics. And because this is I could have shown you other diseases, but I decided to talk about multiple sclerosis because this is a disease where, let's say, the pathology is very localized and it's so you can really define anatomical domains of pathology and non-pathology. So that helps in spatial omics where you can really identify where you have to look. Just to get everybody on the same page, what is MS? So MS is the most common demyelinating disease in humans. A typical age of onset is late teens, early 20s, but the incidence in elderly people is increasing. It is characterized by loss of myelin, and that loss of myelin leads to axonal damage, and that at the end of the day leads to neuronal loss, and that leads to increased disease burden. The typical disease trajectory is that people suffer from bouts of inflammation where these lesions emerge, they become repaired, and then and that stage of the disease is called relapsing and remitting MS. And then typically at some point the disease transforms into a progressive disease where these bouts of inflammation and repair are no longer present, but there's progressive decline in these patients. So the first phase of the disease, this relapsing remitting phase, can be suppressed by immune therapy. But still, the underlying progression is still ongoing, and we really want to understand what are the molecular drivers of this disease. It's characterized, as I said, by lesions in the brain or in the spinal cord, and a lesion is an area where you have myelin loss, which is depicted in the images you see on the right, and the top row is a staining for a myelin protein. And if the area is white, it means the myelin is lost. So you see lesions where you have myelin loss, and the brown around it is where you have myelin is still present. So on the left of the image, that's where the lesion is. And below is a staining for an antibody that recognizes immune-activated macrophages and microglia. And what you can see is that so the lesions can be sort of characterized based on the degree of myelination and the degree of immune activation. And you see this lesion on the left is called active, and the idea is that this is an ongoing, relatively new lesion where you have demyelination, but the whole lesion is still contains activated macrophages and microglia. The lesion in the middle, you see that this inflammatory response is now more restricted to the rim. The idea is that this is a little bit older lesion that is expanding, and that the damage is done on that rim, and that the core is now is no longer inflamed and also hypercellular. And you can have lesions where there's sort of demyelination persists, the inflammation is gone, and this is an inactive lesion, and that is considered to be an unfavorable outcome. And sometimes you have lesions that become repaired, which I don't show here. And what we really want to understand is why does a lesion emerge? How does it progress? What happens at the rim? Because that's where the lesion expands. And why do certain lesions repair and some don't? Why is that? What is the underlying biology, which we don't understand? And we use So this is what you try to understand. Lesion initiation, progression, and remyelination of MS. It's poorly understood. That's clearly a very spatial process. So so this is a disease where spatial omics is very useful because this is a typically architecturally defined process. And with spatial omics, you can address this. And I will show you examples how we did this. So we started doing spatial omics. I think our first spatial experiment was 7 years ago, I think, using a capture platform with very large spots. So basic. At the end of the day, we never published it because it was very sparse data, spots very far apart. You capture 50 cells per spot. So it was basically mini bulk and a poor man's mini bulk experiment. So we never published it. But at the end, newer platforms came along, and the technology is going very fast, where the resolution in your data is increasing. And here we did a spatial transcriptomics using the Visium platform, not the Visium HD yet, that came after. And the premise is that you have glass slides. These glass slides have probes. These probes are barcoded. You put your tissue on top. You capture the RNAs. You sequence it. And based on the barcode, you can map it back in space. That's how it works. And we profiled a whole cohort of lesions from a whole cohort of donors. And you can then plot. So here you see an example how that looks like. On the left, you see a staining of a lesion. So the whitest bit in the center is the lesion. We captured it. And then on the right, we plot the expression of a myelin gene, PLP1, and you see that PLP is lost in the lesion. So you can sort of reconstruct the pathology of the lesion using RNA molecules. It's published, so I won't go into too much detail, but one of the tricks that you can use is the spatial spots that you use to capture RNA molecules. You can sort of identify using an algorithm called Bayes space. You can determine spots that have similar gene activities, and that way you can define anatomical domains in your lesions and extract genes that are, let's say, abundant in cores, abundant in rims, abundant in the perilesional normal matter, etc. You can identify genes that you suspect might drive lesion progression and biology. And what we found, which was, is that around the rims of active lesions, there were genes that are specifically enriched in the rims of active lesions, lesions that are expanding. And this is spatial plots where you see on the top, you see the lesion type. So the first two columns are active lesions and then two mixed lesions. And then 3 genes are plotted, ApoC1, SPP1 or osteopontin. SPP1 will come back many times, so that's the gene you have to remember. And FTL is a ferritin molecule. And you see that around these lesions, you have, especially the active lesions, you have a rim where these genes are expressed abundantly. So it was the first indication which genes and gene programs are dysregulated around lesion rims. But the point is that if we do all this work on post-mortem brain samples, you only get access to a brain sample once. So by definition, all this data is cross-sectional. So we only see the lesion once, and we only see it from a donor once. So in order to predict how lesions progress, you have to use computational approaches because you cannot follow and profile these lesions in time. In mice, you can do this, but in humans, obviously, you can't. So we do computational tricks to do this. So what we then did is that we went back to our spatial data, where you have all these spatial spots where you captured RNA molecules, and we treated all these spots as if it was a single cell object. And then we did by pseudotime analysis, where you sort of order. If you do this in single cell data, you order cells based on gene activity. Here we did the same, but then the input was not a cell, the input was a spatial spot. And on that platform, each spot contains, let's say, 5 to 15 cells. And then we could calculate trajectories, as you can see on the UMAP on the right, where each dot is a spatial spot, and the lines indicate the trajectories that we find, and then we find 3 endpoints. And just to focus on one endpoint is the subtrajectory number 1. It ends at the top right, and if you then look at genes that are expressed along the trajectory, we find back SPP1, which you should have remembered, and it's expressed at the top right of the UMAP. So this is a gene that is very active at the end of trajectory 1, and if we plot that back in space, we know that the spots that are in that end of the trajectory are coming from the rims of lesions. So we know that in the rims of lesions we have a lot of microglia and they express a lot of SPP1. The point was that these spatial spots are also again mini-bulk, 5 to 15 cells. So we went back to our tissue blocks, we extracted nuclei, and we generated a single nucleus RNA sequencing data. So then we have cellular resolution. So we did this for, I don't remember, maybe 30 samples or so. We did single nuc RNA-seq. We subsetted the microglia, and this is what you get. So this is a UMAP with microglia. We find some 10 different microglial states. And we were not the first ones to do this. So we first looked for states that other people have reported in MS lesions. For instance, these foamies. So these are microglia or macrophages that are loaded with lipids because they eat a lot of myelin. So they're lipid-laden, and they have a particular gene signature, and we find them back. It's this cluster on the left of the UMAP. And here at the bottom, what you see is the proportional distribution of these cells across lesion types. So you go from control white matter to the right, and you see that the peak is in active and mixed lesions. So lesions have a lot of these mixed lesions, and active lesions have a lot of these lipid-laden microglia. So this was sort of a sanity check for us. You would expect them to be present in lesions where there's a lot of myelin uptake. You would expect macrophages and microglia that are lipid-laden, and that's what happened. If you then look at this top cluster in green, we call them responsive because they are different from these lipid-laden microglia. And one of the marker genes, again, was SPP1. As you can see on the top right, if we plot SPP1 and other marker genes, it really labels that particular cluster. And then we went back to our spatial data. We can plot then gene modules in our spatial data and see where does this cluster land. And if you do that, it lands, you can see that, for instance, in the middle column. You see that's where in these lesions, you see that the two middle columns, you see that these responsive microglia really decorate the rim of MS lesions. So this is how we go back and forth between spatial data, single nuc data. We calculate module scores. You can go back and forth in where cells that you identify in a single nuc object land in a spatial object, even though it's not single cell resolution. Right? But then the next step for us is, and that's what we're at the moment doing a lot, so the Visium data is, we did this because we had no prior knowledge. We did unbiased capturing where you go genome-wide. We have single nuc objects, and then you can sort of play around with this. But as time progressed, and I already said that technology is going very, very fast, so now you can do this as single cell resolution. I'll move a little bit away from MS, but I'll go back to it later. We are interested in microglia at single cell resolution in space in relation to pathology. A little sidestep. Many labs, including us, have been profiling microglia in the context of disease, in humans or in mouse models for disease. People have looked in mice expressing A-beta for Alzheimer's disease, ALS models, you name it. And what you can see here is that people have profiled microglia in young and older animals, in humans, and what you basically see is that there's a whole bunch of gene signatures being published, and we are guilty as charged, we also did this, and they all have different names, but if you look at the overlap in genes, you see that many genes are induced in these different microglial states that are shared. So we thought, well, maybe there are not 15 different microglia states that are disease-specific. Most likely there's commonality in these signatures that are being induced. So what we did is we went back to our published data. So we worked on Alzheimer's disease. I didn't show that. We have data from MS, which I showed you. We have data from frontotemporal dementia, which I also didn't show. And you can look them up. They're published here. You can see here below. And we basically pulled all the data, which is roughly speaking 200 brain samples from 100 donors or so, and we extracted the microglia and we integrated it. And this is a UMAP of that integrated object. This is all unpublished, which is roughly 400,000 microglia, and we can identify, it's arbitrary, but we can identify roughly 8 states. And what we did is then looked at the marker genes of all these clusters, and we annotated them based on what we think they are doing. Some are disease-associated, some are more inflammatory, some are homeostatic, meaning resting, or what microglia should be doing. A small cluster on the top left, they're proliferating, but we find 2 clusters that are disease-associated, 5 and 8. And the next question was, this is all single-nuke data. We have no spatial information. Can we map these clusters back in space? That is the question. So are these different microglial clusters that we identify, can we map them in space and across CNS diseases? So what we did is that we used a different platform, because now we have a lot of prior knowledge. We have a Xenium machine in the lab, and we designed a probe panel and the idea behind that probe panel is that it reports on all of these clusters that are in this object. So what we do is we do spatial omics using the Xenium machine on CNS tissues from people that suffered from Alzheimer's disease, MS, FTD, you name it. And then we use probes. We use 2 categories of probes. One category is a probe set that recognizes all CNS cells as beacons to identify neurons, oligos, astros, you name it. So we can sort of define the CNS landscape. And on top of that, we have a probe set that recognizes all these, at least presumably, recognizes all these different microglial states. That's the strategy. Now, the output of the Xenium is this. So what you see here is the output of the machine. Each dot is a transcript that you detect. And if you zoom in, all these blue things are nuclei. And now the problem emerges, how do I know which transcript is in what cell? So if you want at the coffee, we can have a discussion about this. It's a pain in the neck. The bottom line, so if you do the most crude way of segmenting, you can say, I only trust the transcript that is on top of a nucleus. That's the most simple way of segmenting the data, but then you throw away a lot of stuff. And since we're greedy, that's not what we went for. We went for a different tool. So I'll explain that in a second. So if you segment out the nuclei, you can then label them based on the genes that are expressed in each nucleus. You can annotate them, give them particular cellular identities. And we did that, and to convince ourselves that is correct, you can plot them back in space, and then they land in domains that are correct. So oligos are in white matter, the neurons are in the cortical layers, astrocytes, microglia everywhere. So we can segment out, based on nuclear segmentation, we can segment out cells, and their identity is correct. And so we do this, and in the paper that I just showed you, how do I go back? It doesn't matter. So we published a paper where so we have a pipeline in the lab where we can do nuclear segmentation. But on the Xenium slides, we can then do antibody staining for markers that we like. And then we integrate the spatial data with the immunohistochemistry data. And we integrate the 2 objects, and then we build AI-aided segmentation tools that we can segment out cells that we like. So we get way more information, because you get way more transcripts annotated to your cells. So that's what we did. If you want, we can talk about this in a minute. All right, so as an example how this works, so this is an MS lesion. On the left, stained for PLP, so this is the... Oh, this is the here. This is the lesion. You see, you have loss of PLP, a myelin stain, and here is a staining for HLA-DR, which is a marker for macrophages, microglia, and you see here the rim of the lesion. So this is a typical mixed lesion. The core is demyelinated, and the inflammatory activity is restricted to the rim. And then these blocks on the left indicate the domain, the area of the lesion that we loaded on our Xenium machine. So we do our Xenium, we do our spatial transcriptomics, we segment out nuclei. The nuclei are then a single-cell object, and then you can plot them back in space. And that's what you see on the right. So you see the UMAP landing in space. And the bottom line is that cells land where they should be. Oligos are in the myelinated part of the lesion. The yellowish cells are microglia, and they decorate the rim. So this is how you can go back and forth between segmenting cells out and checking in space if they land in the correct domain. But that does not answer the question. So So that is just for us to prove that we are able to segment the cells and give them the identity that we think they are. The next thing is, are we able to map different microglial states? That was the whole premise of this exercise. So what we did is then we went back to the spatial data. We did this for controls, for gray matter, white matter, MS, Alzheimer's disease, and different ApoE variants, FTDs with different genetic backgrounds. So we generated the spatial data, we segmented out the cells, and then we see where do they land. So this is a UMap of the microglia that you can find in your Xenium data. We find 6 clusters. The reason that the resolution here is less is that in single nuc RNA-seq, you lose spatial information, but you have way more resolution in your data. Here, the resolution is determined by the number of genes that are in your probe set, and that makes the granularity of the data less high. That can maybe be solved in the future, but at this moment, the probe sets, the sizes of that are somewhat limited. Okay, so as a sanity check, on the left is the same lesion. If you plot a myelin, you see that we use that to delineate the lesion, which you can see here. This is the lesion, this is myelin. And then you can see that there's one microglial state that decorates the rim going inward to the lesion, and there's a microglia state that is on the rim going outward. So we can separate these microglial states, and they have different spatial anatomical restrictions. And then we went back, so there's now tools available where you can sort of integrate spatial data with single nuc data. We have a lot of single nuc data with high granularity and high resolution. We have spatial data. And then we checked, are the microglial states that we map in our single nucleus object, and we made these probe sets for, are they indeed recognizing the microglial states that we mapped in space? Does that work, yes or no? So we used a tool for that, which is called Tangram, where you can basically, I will not go into the technicalities, but you can map your single nuc clusters in space. And that worked. So we find, we find a disease cluster that we identified, which decorates the inside of the lesion, and another disease cluster that we find in our single nuc object decorates more the rim and the outside. The color is not so great, but it's more on the rim going outward. So that convinced us that with this probe panel, we indeed were able to capture the disease states that we were able to find in our single nucleus object. So in a nutshell, what I try to share with you today is that we can map disease-associated microglial states across CNS pathologies. And the take-home message is that these signatures are shared. It's not that each disease has its own private signature. That's not how it works. They are shared. And if you then map these different disease states, they occupy different anatomical domains. We also have this we know this is true for AD, I didn't show you. We see that they associate with amyloid or with other pathological features. So in MS, it's very clear because you have this very clear demarcation of the lesion. So we have data now in the pipeline where we can map different microglial states to different pathological features in space. I try to give you some insights on how we use omics to understand what microglia do, and how you can use different approaches. I did not talk about long reading and whatnot we are doing at the moment, but anyway, this is sort of I hope I could convince you that looking at RNAs can give you insights in what's going on in CNS pathology. And this is my most important slide. So these are most of the people in the lab. On the top right, you see the current members and the staff members at the top that all have contributed to this in many ways. In white, all the postdocs and grad students and PhD students and techs. I try to acknowledge people at the bottom who contributed to published work, and at the bottom, the sponsors that made this work possible. And thank you for your attention.